Hello, everybody. Greetings from Healthcare Foundation of India. I'm Dr. K. K. Agarwal, President, Confederation of Medical Association of Asia and Oceania, past National President, Indian Medical Association, and President, Healthcare Foundation of India. And you are watching Medtalks with Dr. K. K. This is going to be 210th episode, 1679th COVID episode. Let's have the headlines of the day. Indian nationals are advised against visiting Dubai consulate amid spike in COVID-19 cases in the city. A total of 2,072 Indians died of coronavirus infections abroad, as per the government sources. Schools in West Bengal reopened for class 9 to 12 after a gap of 10 months. The first batch of Pfizer Incorporation COVID-19 vaccine arrived in Japan on Friday, local media reported, with official approval for the shots expected within days as the country races to control a third wave of infection ahead of the Olympic Games. US acquires 200 million new doses as the vaccine drive begins in pharmacies. Let's have the topic of the day. Testing for neutralizing antibodies. The new test, which we announced a couple of days back, has been introduced in India. Before I introduce my special guest, it's customary in our country, Atithi Devo Bhava. Atithi Devo Bhava means Suman ke, we wash our hand, S for Seedha, U for Ulta, M for Mutti, A for Angutha, N for Nakhun, or K for Kalai. Once we are ready, let's introduce our special guest, Dr. Vandana Lal. She is the Executive Director of the Lal Path Labs, India's #1 pathological lab. She has been awarded the Women of the Year Representing India Award by the American Biographical Institute in 2001 and 2008. Superheaded by Dr. Vandana Lal, the unit has done India proud by developing highly sophisticated tests in molecular diagnostics. You cannot think of Agra without Taj Mahal. Similarly, you cannot think of laboratory without Dr. Lal Path Lab. I also have with me Dr. Salab Malik. He is the National Head Microbiology and Serology Molecular Infection. He is an alumnus of LTM Medical College, Mumbai. He is practicing clinical microbiology since last 20 years in Delhi. He has been associated in past with Quest Diagnostics, Columbia Asia Hospital, Paras Hospital, Primus Hospital, Shri Balaji Action Hospital, and visiting consultant to many reputed labs in South and West Delhi. His areas of interest include lab automation, antimicrobial resistance, mycobacteriology, and quality and management issues in laboratory. First of all, I like to interact with Dr. Vandana Lal, and then we will listen to Dr. Salab Malik, and then we will be back interacting with them on question answers which you may have. This program is live on 5 different platforms, and we expect a large number of people interacting and understanding the concept. Dr. Vandana, why do
Good
you
e-
think that bringing neutralizing antibody test was the need of the day?
Introducing the neutralizing antibody test is required because this is the test which will actually judge how the immunological response is in a person, especially following the vaccination. The other kind of antibodies which we were testing earlier, like IgG and IgM and total antibodies, those antibodies were towards the entire surface of the virus. Whereas the neutralizing antibody is specific to the spike protein, which is causing damage to the cells in the body. So it was very important to introduce neutralizing antibody because these are the antibodies, as the name suggests, which will neutralize the effect of the virus and hence to study the effect of the vaccination in individuals, especially now that vaccination has started in India, it is very important to assess them by doing this specific test for the coronavirus.
At what interval I should expect that some degree of neutralizing antibodies have started after the first vaccination? We know that normally we check 2 weeks after the second vaccination, but do you think something will come at the third week of the first vaccination?
What is recommended as per the research which has been done in this field is that after the first dose of vaccination, 2 weeks after that, we should check. And after the 2 weeks after the second dose, and then again at 3 months, what is recommended? It is expected that the neutralizing antibody will last for 6 months. However, the disease is evolving, and as more and more research papers are available, we will know the longevity of this neutralizing antibody.
Is there a cutoff value where we say good, bad, best, better?
There is it is cutoff is only for positive or negative. However, the higher the value of the antibody, that means the better is the immunological response in the individual. What we have seen is that people who are asymptomatic or who have very mild infection, the level of antibody in these individuals is rather low. But people who have had a severe form of COVID-19 disease, those individuals generate a higher level of antibody response. The only exception to this rule is people who are on immunosuppressants, or who are suffering from HIV/AIDS, or whose immunological status is depressed due to certain medications. Those individuals do not show a good immunity response.
So, as on today, because after the first vaccination, people have started getting now the second vaccination, what are your preliminary studies about these vaccinations? Are post vaccination antibodies, are people showing them?
Yes, people are definitely showing new antibody levels after the first dose of vaccination also. Majority of the people are showing. However, there is a certain population in which we have found that 2 weeks after the first dose, they do not show a good response or may not have any response at all. But however, the second dose is still to be given to them. So we are following up on these patients.
Do you feel in future, Dr. Lal Path Lab will also bring a cell-mediated evaluation of a memory T cells and memory B cells? Are you also working on that?
We are working on it. We are waiting for kits to be developed. As you know, the disease is evolving. The vendors are manufacturing kits as per the research papers which are developed, as well as the research in their own organizations. So, since the disease is just a 1 year old, it will take some time for these kits to be available commercially in the market for testing. However, we will be the first to introduce any kind of cell-mediated immunity when moment the kit is available.
So, as on today, you feel that because most of the healthcare workers have received the vaccine, and now we will start getting people 50 plus who will need the vaccines with that. So, your recommendation is 2 weeks after the first vaccines, 2 weeks after the second vaccine, and at 3 months, we should get a neutralizing antibodies test done.
Yes, that is the recommendation as of now. And also, what we are waiting for is for the more and more people to get vaccinated, because if 50 to 60% of the population is vaccinated, we assume that herd immunity would have developed in India. And these are very preliminary figures, and some research has been done in the National Institute of Health, NIH, in America. And in fact, certain countries, certain governments are promoting that we should issue an immunity passport now. However, World Health Organization, WHO, is not in favor of this, because as of now, we know that these neutralizing antibodies, even if they are present in a person, what are the chances of reinfection is not yet known. So it is important to be vaccinated, important to be followed up with neutralizing antibodies for the safety of the individuals.
Anything else before you ask Dr. Shalab to present? Anything else you want to say about neutralizing antibodies?
Uh, Neutralizing antibodies are the most specific kind of test which is available at the present moment. And this is the most specific test because it shows that these are the antibodies which are actually killing the virus in the body. The other antibodies which we have studied, like total and IgG and IgM, those antibodies are very non-specific towards the virus and may not protect you completely. So it's very important to do the neutralizing antibody, which will neutralize the effects of the virus. However, now I hand you over to Dr. Salab Malik, who is an expert in this field and will make a presentation regarding the importance of neutralizing antibodies.
Before Shalab takes over, how many centers you have in the country?
We have 210 labs all over India. We have a lab in Bangladesh and Nepal also, but we have nearly 1,500 collection centers and 7,000 pickup points from where we are getting samples from all over India.
And you feel that this test will be available pan-India?
Yes, that is our aim, to introduce it all over.
So that anybody who is watching this program, not necessarily in Delhi, anywhere, you can ask for this test. And I've given to understand that you collect samples up to Thursday, 5 o'clock, and then then you do the test in bulk. Am I
Yes,
correct? Or
yes, that is correct. And we have started only in Delhi, but gradually our aim is to make as many centers as possible so that there is easy accessibility to the public.
okay, now let's listen to Dr. Shalab Malik, the scientific aspects and some of the preliminary data, and then we'll come back for discussion. Over to you, Dr. Shalab Malik.
Thank you, ma'am. And
You can show the slides. Kindly go ahead. Your slides are visible.
yes, yes. Is my screen visible?
Yes, yes.
Okay. So today we will be talking about SARS-CoV-2 COVID-19 neutralizing antibody test, which is latest in this series. And before I begin the actual presentation, there are many questions in our mind. What is the significance of this neutralizing body, and what is their role in different strains, genotype, etc.? How different is this test? How is the kit different from other serological kits available in the market? What is the mechanism of this test? Then how do you apply this test? Whether you will use it to ascertain the herd immunity, or in clinical therapeutics, or in monitoring the vaccine efficacy, and so on. And finally, what is the role of this test in immunocompromised person? Will it work where some degree of immunocompromised status is there, and how it will progress? So these are the common questions which I will try to answer in this presentation. So the pathway of this presentation will be starting from the virus structure. Then we will be talking about mechanism of infection, test and timing. And CPAS is the SARS-CoV-2 neutralization antibody kit, and how it is different from the traditional IgA, IgG, IgM assays. And finally, what is the semi-quantitative workflow, and what happens to the cross-reactivity with this kind of testing. So, first of all, why do we need such a test? Although so many IgG tests are there, there is nucleocapsid antibody testing going on, spike protein antibody testing is there, and off late there was rapid cards also, IgG, IgM, and IgM never took off in India. So, what was the need to get this? Because we noticed some false positivities was there. Persistence was the issue. We were not confident to say that, okay, now your IgG is detected and you are 100% immunoprotected and you will not get the virus, and this antibody will be good enough to block the entry of virus. So with the existing antibody test, we were not sure to answer this question. That is why nobody was committal. That you show the IgG and it becomes your immunity passport. So that kind of confidence was lacking. So is it a neutralizing or just another antibody? And there was a lot of confusion, and this confusion was super added with the rapid kits also at one point of time, which was finally banned in Indian territory. So after four antibody tests, I am still not sure I have got COVID 19 or not. And this is the first time in the history of viral disease where the science was not confident to advocate it for diagnosis of the test. And all the antibody tests were restricted to the serosurveillance. So there was continuous notification from ICMR and Government of India side that please, for heaven's sake, don't use the antibody test for diagnosis. Its intent is to just monitor the serosurveillance, and nobody was committed at all on the immunity aspect of this antibodies. So definitely we need a better kit. Now almost 10-11 months are over, and we have come to the conclusion that just detecting the antibody is not good enough, as all antibodies are not equal. They are responsible, they are coming out of the different isotypes, different serotypes. So there are a lot many questions. So with neutralizing antibodies, finally we are able to pin down those questions and give you a good answers. So first of all, let us understand what is a neutralizing antibody. A neutralizing antibody is a type of antibody that is capable of keeping an infectious agent, a virus in this case, from infecting a cell by neutralizing it or inhibiting its biological effect. So either it has to block the entry or just inhibit its multiplication further. So that is the main idea of neutralizing antibody. And so far, not only in this disease, we have been seeing this neutralization effect in hepatitis A and B and influenza, then hepatitis B, especially now we have got a good experience by monitoring it with anti-HBs titers also. So that is on the higher side of the scale. So this is once again a quick diagram that first 8 days, first 7 days, PCR has got the role to play, which will be mainly diagnostic in nature. And one fallacy of PCR is it cannot tell you what happened previously. So it can answer the question in the current context of the clinical situation. Then comes the antibody test, as you see on the screen, that 14 to 21 days definitely serological positivity starts developing, and then
So next
they
slide.
try to,
Next. Yeah.
yes, uh, they try to be present for 6 months as of now, whatever data is available with us. In case of IgM, they have noted that these antibodies perish after 35-40 days. In case of IgG, 3 to 6 months, they are saying. So that is why quarterly evaluation or a 6 monthly evaluation will be the need of our as far as serological tests are concerned.
Med-
So one thing is clear that so far, whatever antibody test we were talking about, they are also called binding antibodies. So all binding antibodies which you detect does not translate into neutralization. So that is why I have put a not equal sign over here. And neutralizing antibodies only result in the protective immunity. And this is a comment from a Moderna vaccine trial also, which says that the thing has made possible everyone be cautiously optimistic is that we did not just see binding antibody, it was clearly antibody that was neutralizing the live virus at the levels that would predict the would be the protective if in fact the neutralizing antibody, which is a reasonable assumption. So before I explain the further mechanics and how this test is superior to the existing test, let us first understand the structure of this spike protein, because throughout our discussion and so far you must have heard about different vaccines also. So it is the spike protein, because this is the one which is actually important in attaching with the receptors which are present in our cells to gain the entry. So look at this figure. The orange thing you are seeing and the yellow bar which you are seeing here, this is the receptor binding domain. Now this is very important. And in this essay, what we have done, we have tried to isolate it and purify it and make a separate concentrate of this. And this type of purification is not at all available in the existing antibody test, where it is taking into account the whole molecule. Now, this is the full spike protein from orange to the yellow. And the main principal one is this yellow one. This is RBD, also called as receptor binding domain. And look at this blue thing. This is a ACE2 receptor. Now this RBD has to attach with the ACE2 receptor to gain entry. And in neutralization, what will happen? This will be blocked. So, and how we will block it and simultaneously detect it on a ELISA plate, I will show you further in the presentation. So once again, this structure, this red color are the neutralizing antibodies. Now this thing, mushroom-like thing, swollen thing which you are seeing is the RBD, and this antibody will get attached over here. So with the result, this they will not be able to combine with the ACE inhibitor, ACE2 receptor, and finally gain the entry inside the cells to give you a cytopathic effect development. So generally, they start appearing somewhere around 5 to 10 days post infection and peak around 20 to 30 days. So as KK sir was asking, which will be the best time after vaccination to monitor this? So somewhere around third week will be good enough to one can subject to this kind of testing. And so far, whatever data is available with us, 6 months longevity has been demonstrated for this neutralizing antibody. Compared to the binding antibody, which we were so far talking, they perish earlier than the neutralizing antibody. So once again, a quick recap of the nucleic acid as well as serological testing. And if you refer to the Infectious Disease Society America document, they have okayed it for the application of this antibody test in diagnosing the infection also, especially in those situations where clinically you are very certain and repeatedly PCRs are coming negative. So this can definitely aid in the diagnosis. But as far as Indian government and ICMR recommendation is concerned, antibody tests are totally no-no when it comes to diagnosing the COVID infection or even as a complementary aid to the COVID infection. So current available technologies are this. Diagnostic is PCR and antigen based test. Then we had binding antibodies test, that is IgG, IgM. Again, they were targeted to different type of antigen. There was nucleocapsid in some of the machines and some of the kits. And there was RBD based technique in some of the kits, and spike protein based technologies in some of the instruments and kits for this thing. IgM, as I said earlier, also it never kicked off in our country, so we don't have much of experience with IgM. And latest is the viral neutralization test, and they become very relevant when it comes to assess the post-infection scenario, especially when you want to know, okay, now antibodies are there, and what is their capability as far as neutralization or inhibiting the attachment of virus with the cell is concerned. So what are the pre-existing neutralization tests and what is a conventional neutralization testing as far as viruses are concerned? Now, as you can see on the screen, existing ones are virus neutralization test. That is a very conventional one. Now here you deal with a live coronavirus, that is SARS-CoV-2. These are the spike proteins, and you will incubate this live virus with the patient serum, whom you are supposed to detect the neutralizing antibody. And finally, this cocktail is poured on the cell culture plates, which are laden with the ACE2 receptors. and there is of course an indicator dye, or you can go for the visualization of plaque development or the cytopathic development of the cells under the microscope. So problem over here is that this will require a highly specialized unit, and it will require BSL-3 facility as you are handling a live virus. Then you will be getting answers in 2 to 5 days. Plus, consistency is a problem, infection hazard is a problem, and scalability is not possible because, as you know, the viral culture labs are limited, and targeting the mass population where you want to find out the neutralizing ability, it will not be possible with this type of testing. Then comes the pseudovirus based virus neutralization test, which we call as PVNT also. So here instead of coronavirus, we choose a lentivirus on which these proteins are expressed. So these are the spike proteins. So once again, the mechanism is same. You incubate it with the patient serum, and if neutralizing antibodies are present, they will not be able to attack the ACE2 receptors on the cell lines, and finally there will be no signal development, and that will tell you that okay, the neutralizing abilities have been detected. But if it over here, like antibodies were there, in this case antibodies were there, they were not able to attach, so no signal development. And in this case it was able to attach, and that is why the signal development, and thereby the things were possible to detect. And finally, the new test is the SARS CoV-2 neutralization antibody test. So here you don't require the hi-fi BSL-3 labs and all. A good biosafety cabinet with a BSL-2 lab is good enough, and you get a quick answer in 1 to 2 hour time. So this is just the gold standard test. They were talking about a live virus-based virus neutralization test, which we have discussed how the it is tedious and complicated to do. So coming back to our test, that is surrogate virus neutralization test. Now what we have done, so as you see this yellow color cap, this is the RBD domain. So this has been purified and combined with enzyme, that is horseradish peroxidase, and the solution has been developed. So what we do, we mix the patient serum with this RBD concentrated solution for almost half an hour to 1 hour, and then finally this mixture is poured on the ELISA plates, which are laden with this ACE2 receptors. So you can see this violet color things. These are the ACE2 receptors. So look at this scenario. If neutralizing antibodies are present, they will immediately bind the RBD molecule over here. And finally, when you will put the mixture on the plate, nothing will be available to attach with this ACE2 receptors. So that means the neutralization has been detected. So two things have happened. Not only you have pinned down the RBD area, but also you have demonstrated the final attachment with the ACE2 receptor. And if no neutralizing antibodies are there, then the RBD area is free and it will interact with the ACE2 receptors over here. So it does not require a high-end biosafety features like BSL-3 of a virus neutralization test. It is fast and automatable and can give you a high throughput. You get a quick answer in 1.5 to 2 hours, and scalability is possible with the surrogate virus neutralization test. So this is again the final mechanics of this. This green color thing is again the ACE2 receptors which are present at the bottom of the plate. Red one is the neutralizing antibody. This bluish thing you are seeing is the non-neutralizing antibody. Now this is the RBD thing which has been purified and made into mixture. And this is how it happens. If no neutralization is there, you get a good color. It works other way around. If you get neutralization activity, you don't get a much of a color when you finally put a TMB dye at the end. So then comes the next question: how good is the concordance or consistency when we combine with the existing assays, that is the nucleocapsid based or RBD based or spike protein based antibody tests, which are already available with us. So no, just let us have a look over here. So, as compared to the other test, the when I talk about this neutralization test, we see the accuracy of around 99.2%, positive predictive value of 100%, negative predictive value of 99%. If I look at the RBD coated plate, that is the one which I was talking about, the binding antibody detection test. So accuracy in that case was 94%, positive predictive value was 64%, negative predictive value was 99.59.
Next
And
slide, please.
yes. So next question comes to our mind that what happens to the cross reactivity? How much cross reactivity was studied? So
Next.
Next.
if you if you look at the screen, you are seeing that the cross reactivity was established by the vendor for influenza A, influenza AB, RSV, hepatitis viruses, then other coronaviruses, and finally, in case of SARS-CoV-1, that was responsible for the 2003 SARS infection. There they found one case positive. Rest all the other viruses antibodies were not at all reactive with this. So again, this can be easily ruled out because now we are dealing with SARS-CoV-2 infection, and looking at the geographical extent of our country, hardly any SARS-CoV-1 is there at the moment. So this can be easily ruled out and not much of a big problem over here.
Next.
So coming to the efficacy, coming to the utilization or scope of this neutralizing antibody-based testing. So one is of course serosurveillance, because after all you have detected the antibody. So it has shown a good agreement with most of the PCR given positive cases. One was able to pick up the neutralizing antibody. So good tool for serosurveillance. Then enable drug identification. If somebody is working on antivirals and all, and you want to examine the ability of drugs or convalescent plasma to block RBD ACE2 interaction, because what was happening earlier, that though you detected antibodies, nobody was committal that you can be a good plasma donor, and you must have seen the cases where the person has given plasma showing the presence of antibodies and nothing happened, because we were not good enough at that point of time to demonstrate the neutralizing ability of that plasma. So everybody was guessing. FDA gave for couple of kits, okay, for Johnson and Johnson kit, 12 signal value will be good enough. Then somebody came out with a diazorin thing, and they said 1 is to 160 will be good enough value for this, but nobody had the concrete answer at that point of time. So this thing has been solved by neutralizing antibody test, especially in ascertaining the value of this plasma donor. Then what happened when we compared it with the conventional viral neutralization test? It showed a good consistency and good agreement with that. So a good test to monitor the vaccine efficacy, and which I am very sure that in coming few months, this data will start emerging, which we will be able to share with you. And finally, it definitely screens the community for neutralizing antibodies, because so far all the tests that has been done to answer the presence of antibodies. So with this add-on, you will be very sure that okay, now in this particular population, the neutralizing antibody has been detected, and herd immunity is very confidently commented upon. So once again, even FDA has okayed it. These are the for vaccine efficacy, convalescent plasma determination, and therapy validation. So this FDA approval is there for this test. So finally, this was
Next.
of the journals that Duke News Gene Script, which says that the world has finally got a test without the need of containment facility or actual live virus to demonstrate the neutralizing ability. So this is the binding antibodies decline more rapidly than the neutralizing antibody, which I had said earlier also. So so far the data available with us is the 180 days they are saying, and we will see in the future course that how long these neutralizing antibodies are there, or will it totally change the pathway, which will require revaccination, as we see in the case of H1N1.
Next.
So now, coming to the final sensitivity and specificity using the plaque reduced neutralization test, that is the conventional viral neutralization test as a comparator. Both FDA and European Union have approved this test. And what we have seen is the negative percent agreement is 100%, and positive percent agreement is also 100%, especially when the plaques were above the 80 or 90 cutoff value. And then there the consistency with the neutral convention and this surrogate neutralization test was good.
Next.
So, this is the final this thing about the comparative figures when we talk about the RT-PCR given positive
Next.
cases. So as you see on the screen, that SARS-CoV-2 positive 47 cases, negative 8 cases. Neutralization test was able to demonstrate the antibodies in all, and in negative, no demonstration was there. So, leading to the final specificity value of 99.1%.
Next.
So finally, coming to the last few slides, that is the test details, which is my screen is showing that the code of this test is S290, and
Next.
we do it from serum, and we require one SST to carry out this test. So this is how the report will emerge on the finally, that it will comment on the negative and positive values, that okay, neutralizing ability has been detected. And as far as FDA is concerned, they have kept a very high threshold of 30%. So it has to cross the border of 30% before the positivity is reported. Now, next question in your mind will be what about the quantitative values and all? Well, they are still working, and I think by March they will standardize it, where they will be confident enough to tell you how much international unit are good enough for this thing. So, thank you for patient listening, and this is my contact detail.
Can you go back to the last slide?
House is open for the questions.
So all those who are watching this program can take a print screen of this, and you can have the queries, and you can have email addresses. People can do that. So coming back to discussion, so as on today, we are doing only qualitative tests and not a quantitative test.
Right.
Okay. So it will, and you are saying that up to 6 months you are seeing. In any way, we don't have the data of 6 months because post-vaccination people have not yet crossed 6 months. But you are assuming कि 6 महीने तक ये antibodies रहने की उम्मीद है. I'm going to speak out in Hindi little bit. So you feel
Yes.
that कि 6 महीने तक ये चलेगा इसके साथ में न्यूट्रलाइजिंग एंटीबॉडीज. नंबर 2. नंबर 3 इज आज की डेट में जिसकी डू यू फील दैट लैब्स एंड हॉस्पिटल्स एंड क्लिनिक्स विल स्टार्ट एडवर्टाइजिंग कि मेरे सारे लोगों को न्यूट्रलाइजिंग एंटीबॉडीज पॉजिटिव है. डू यू फील कि ये सिनेरियो आएगा?
Yes, actually, this is important. Rather than going for the other test, this is the fine. This will be the final immunity passport. That okay, I am because of the infection or because of the vaccination, I am carrying a neutralizing antibody with me will be very important evidence. In
So
fact, there is a commentary also in some of the international journals where Australia has come out very openly for this.
तो अगर मेरे एस एंटीबॉडीज पॉजिटिव हैं रूटीन टेस्ट से, बाइंडिंग एंटीबॉडीज पॉजिटिव हैं
Right.
और मेरा न्यूट्रलाइजिंग एंटीबॉडी नेगेटिव है, तो इसका क्या मतलब हुआ?
उसका ये मतलब हुआ है कि जो एज आई शोड यू इन माय प्रेजेंटेशन आल्सो दैट एस मॉलिक्यूल इज वेरी बिग and actually it is the RBD, that is the receptor binding domain, which is important. तो वो इसका मतलब वो जो है वो ब्लॉक नहीं हो रहा है और दैट इज व्हाई इट इज नॉट एबल टू प्रोटेक्ट यू फ्रॉम एंटरिंग द सेल और प्रोटेक्ट यू फ्रॉम द इन्फेक्शन.
तो
So
इसका मतलब यह हुआ कि वो एंटीडी एंड टर्मिनल डोमेन या नॉन आरबीडी नॉन आरबीएम की एंटीबॉडीज
yeah,
बनी है. दैट्स व्हाट इट
absolutely.
मींस. ओके.
Yes.
तो जो डिसीज एनहांसिंग एंटीबॉडीज होती हैं उसे कैसे पता लगाते हैं?
Disease enhancing nucleocapsid can be one which can tell you about this. But the primary objective of this test will be to decide how much is the prevention possibility or blocking ability of the antibodies there, so
नहीं,
that you don't get the.
कुछ ऐसी एंटीबॉडीज होती है ना, जैसे डेंगू में वैक्सीन दिया था तो डिसीज एनहेंसिंग एंटीबॉडीज आई थी जिन्होंने बीमारी को बढ़ा दिया.
So that happens when they try to become a complement activating type of situation will happen. So rather than giving you protection, it will enhance. So such thing so far is not, no data is available for this type of disease, and we have to see it further. And over here,
तो
one more information I will like to share, that what happens to the different strains. So when this kit was being developed, what they had done parallelly, as you must be aware that there are different genoclades of the SARS-CoV-2. So all the genoclades possible with their respective sera was also tested. So far what they discovered, that these antibodies were equally functional in the different type of strains, and they were not able to find out still yet that strain variation will make much of an impact in
सो
the detecting the neutralizing ability.
सो इसका मतलब है जो 6970 डिलीशन है एंटीडी एन टर्मिनल डोमेन में, जिसका R1 सिचुएशन में, वो तो हमारे को एंजीन फेलियर देता है, एंजीन टारगेट फेलियर देता है इन थर्मोफिशर टेस्ट. लेकिन
Right,
यहाँ
right.
की हम बात कर रहे हैं कि जितने भी आरबीडी और आरबीएम वाले, क्योंकि 501 is the only RBD mutation in UK1, but we have two more mutations, one in RBM and one in RBD in South Africa virus, जिसके अंदर
Right.
में 484 है and 417 है. तो उससे आज की डेट में इस टेस्टों में फर्क नहीं पड़ने वाला, यही कहना हमारा.
हम ये कहना चाह रहे हैं कि जो पीछे के जितने जीनो स्ट्रेन्स थे और क्लेड्स थे उसमें कोई फर्क नहीं देखा गया, क्योंकि ये रीसेंट है सारा, साउथ अफ्रीका वगैरह का ये रीसेंट डेवलपमेंट है. सो वी आर येट टू नो द व्हाट विल हैपन फाइनली. नोबडी नोज राइट नाउ
सो
अबाउट द
अभी
इम्पैक्ट.
जो आप लोगों ने केसेस किए हैं अभी तक, आई एम टॉकिंग अबाउट
राइट.
ओनली दिस पर्टिकुलर टेस्टिंग, जिन लोगों ने टेस्ट कराए हैं उसके प्रिलिमिनरी रिजल्ट्स क्या आ रहे हैं? जिन
See,
लोगों को
so
वैक्सीन
far,
लगा है,
yeah,
वैक्सीन
so far
जिन्होंने
100 odd tests I have done, and we were able to pick neutralizing ability in somewhere around 65 to 70%. And there was one interesting case also. I came across one doctor who happened to get vaccinated in last October itself, and he had taken 3 doses of vaccine. So he got antibodies test done that was negative. So when finally I interviewed him, I came to know that the antibody test which was carried out was nucleocapsid. So then I suggested him go for neutralization test. So last week we did neutralization test for him, and to our good fortune, we were able to demonstrate the neutralizing ability of 89% in
ओके.
that case.
तो हम लोग वैसे जब स्टूडेंट्स को पब्लिक को पढ़ा रहे हैं विच आई एम डूइंग आई हैव डन अराउंड 1680 मीटिंग्स सो फार एंड तो मैं ऐसे पढ़ाता हूँ तो आई वांटेड टू नो मेरा assumption करेक्ट है कि नहीं. I said antigen is like a चोर, एंटीबॉडी इज लाइक ए पुलिस. 3 प्रकार की पुलिस होती है. एक होती है करप्ट जो पिक्चरों में दिखाती है. वो करप्ट पुलिस होती है, वो चोर को पैसे देती है कि तू चोरी कर, मैं खड़ा हूं. वो उसको पूरे
Right.
सीक्रेट्स देती है. सो दीज आर डिसीज एनहांसिंग एंटीबॉडीज. फॉर्चुनेटली कोविड के बाद नहीं दिखाई दे रही कोविड के केसेस में. दूसरी निकम्मी पुलिस होती है जो नॉन न्यूट्रलाइजिंग होती है. वो पुलिस आके खड़ी रहती है कि जा रहा है जाओ, मेरे को क्या लेना देना. और तीसरी सिंघम पुलिस होती है जो कि सिंघम, एवरीबडी नो सिंघम नाउ एज ए पुलिस ऑफिसर. तो वो पुलिस ऑफिसर वो वाले हैं जो जान जाए लेकिन चोर ना जाए, चोर को पकड़ना है.
Yes, yes, well said. Absolutely.
तो ये
Absolutely.
न्यूट्रलाइजिंग एंटीबॉडीज जो है सिंघम एंटीबॉडीज हैं. तो अगर आप चाहते हो आपके शरीर में सिंघम एंटीबॉडीज है कि नहीं है, तो न्यूट्रलाइजिंग एंटीबॉडी टेस्ट कराओ.
Right. And finally, they are actually will not only talk about the IgG and IgM or IgA, they are able to detect the neutralizing ability of all three. So that means this type of testing is independent of the type of antibodies, which was a restriction in the existing binding antibody test. So that has been removed.
जो हमारी क्लास 1, क्लास 2, क्लास 3, क्लास 4 एंटीबॉडीज है, इसमें रिस्ट्रिक्शन नहीं है.
इसमें नहीं है. It will work across. So wherever this binding possibility is there, it will work.
So anything else you want to say? The test is available. Is it a costly test or is it a reasonably placed test, Vandana ji?
Yeah, right now it is a expensive test, and but I am very hopeful with the passage of time, the cost should come down, and also we will be able to do it on daily basis. So far I am doing it once a week, and as ma'am has already said that parallel validations are going on at our different labs. So very soon it will be available across the country at different facilities.
So
And
just
what is the
like the
cost?
COVID test, just like the COVID test which we started, and it was priced at 4,500, and gradually, it came down to nearly 800 rupees. Similarly, as the vendors are able to sell more commercial kits, the price is definitely going to come down.
The current price is?
Current is around 4,000.
Which is okay, which is, you can say, whenever a new technology starts, so it is at the same price as which RT-PCR started.
Right.
So you can say it is cheaper than the original RT-PCR price.
Right.
And
And
with
we hope
time,
it will
the
come
price
down.
will go down.
Yeah,
Yes,
it will
with
go down.
with the things it will come down. So
Right.
so I think जितने भी लोग इस प्रोग्राम को देख रहे हैं, if you really want to know the protection, you need to have at least one test, if not 3. And if you want to have a one test, then do it 2 weeks after the vaccine. And if you can afford, you can go for 3 testing. But definitely एक बार तो टेस्ट अगर आपको मालूम होना जरूरी है कि मेरी न्यूट्रलाइजिंग एंटीबॉडीज है कि नहीं है. स्पेसिफिकली इन हाई रिस्क एरियाज, घर के अंदर में हाई रिस्क एरियाज हैं. मैडम ने कहा है और शलभ जी ने भी कहा है कि आईसीएमआर कहता है डायग्नोस्टिक में एंटीबॉडीज का कोई रोल नहीं है. लेकिन क्लिनिकल प्रैक्टिस में हम सारे क्लिनिशियंस एंटीबॉडीज के साथ में एंटीजन को इंटरप्रेट करते हैं इन डिफिकल्ट कंडीशंस. क्योंकि अगर हमारे को न्यूट्रलाइजिंग एंटीबॉडीज पॉजिटिव आती हैं, तो फिर हमारे को, अगर हम किसी कोविड-19 से एक्सपोज भी हो जाते हैं, तो हमें क्वारंटाइन में जाने की जरूरत नहीं है. दूसरा, अगर हमारे को न्यूट्रलाइजिंग एंटीबॉडीज पॉजिटिव हैं, अगर री इन्फेक्शन हो भी जाता है तो सीवियर नहीं होगा, सीरियस नहीं होगा, हम मरेंगे नहीं. एटलीस्ट एक श्योरिटी आपके पास रहती है. तीसरी श्योरिटी है, अगर आप न्यूट्रलाइजिंग एंटीबॉडीज का सर्टिफिकेट लेके फॉरेन ट्रेवल कर रहे हो, तो आप वहां जाके ऐसी नौबत नहीं आएगी कि वहां खांसी जुकाम हो गया और आपको 15 दिन के लिए बंद कर दिया. Because that can happen. You can acquire a simple flu in a foreign country और
Yeah.
वो आपको सीधा आपको होटल में बंद कर देंगे. So if you are carrying a certificate of a neutralizing antibodies, definitely सरकार माने ना माने, FDA माने ना माने, बट ये इनडायरेक्टली एक तरीके का इम्यूनिटी पासपोर्ट है. Binding antibodies, people were not thinking it as a immunity passport, but neutralizing antibodies, yes. So whenever some doctor is asking you मेरा antibody test करा दो, तो पूछो कौन सा antibody test करा रहे हो, binding या neutralizing. Binding का मतलब है वो वायरस को पकड़ लेगा लेकिन मारेगा नहीं, जिससे मैंने बोला है निकम्मी एंटीबॉडीज, निकम्मी पुलिस. वो चोर को जाके पकड़ लेगी लेकिन उसको अगर चोर छुड़ाएगा तो छोड़ देगी. लेकिन अगर आपकी न्यूट्रलाइजिंग एंटीबॉडीज हैं तो फिर वो वायरस को अंदर घुसने नहीं देगी. याद रखिए आप सब लोग, खासकर क्योंकि पहले फेज के अंदर में हेल्थ केयर वर्कर्स को लगा है. आई होप डॉक्टर पैथलैब विल गिव some special offer for the doctors and the healthcare workers, and get your antibodies done. आज नहीं तो कल, I am 100% sure all doctors, हम इंडिया में 700 डॉक्टर्स को लूज कर चुके हैं. सारे डॉक्टर्स जानना चाहेंगे कि मैं प्रैक्टिस में जा रहा हूं, मेरे को रिस्क कितना है. उस सिचुएशन में अगर न्यूट्रलाइजिंग एंटीबॉडीज आपकी पॉजिटिव आती हैं, एटलीस्ट यू विल बी श्योर कि इन्फेक्शन होता है तो होए, आई डोंट केयर, ना मेरे को सीरियस बीमारी होने वाली है, ना मैं मरने वाला हूं. दैट्स व्हाट वी वांट फ्रॉम द पीपल. लेट मी थैंक डॉक्टर वंदना एंड डॉक्टर शलभ टू बी हेयर. बट जाते जाते 3 टेक होम मैसेजेस, 3 डॉक्टर वंदना से और 3 डॉक्टर शलभ मलिक से हम लेना चाहेंगे कि आप, वंदना जी, क्या चाहती हैं डॉक्टर्स ये 3 पॉइंट जाने, एंड डॉक्टर शलभ मलिक पब्लिक इसके बारे में 3 चीजें क्या याद रखें. वंदना जी.
So for the doctors, I would say that if they have to do the test, they should do only neutralizing antibody test to check the efficacy of the vaccine if they have been vaccinated, or to check if they have suffered from the disease that they have developed neutralizing antibodies. Second point I would like to say is that if they want to do one single test of neutralizing antibody, then ideally they should do it after 45 days of vaccination, so that both the doses have been given plus 2 weeks have elapsed after the last second dose of vaccine, so that they at least we see the presence of neutralizing antibodies. And the third thing is that it is as the paper research work happens and more information is available, we will be able to participate again in this program with Dr. Agarwal and give you latest information on the neutralizing antibody. And I hope Dr. Agarwal will accept our wishes.
थैंक यू.
Thank you so much.
शलभ जी, पब्लिक को क्या मालूम होना चाहिए, लेकिन एक चीज है वंदना जी, अगर आपने पहला टेस्ट पहले वैक्सीन के 2 हफ्ते बाद करा लिया और पॉजिटिव आ गया, तो फिर बाकी 2 कराने की जरूरत नहीं है, क्योंकि अब वो डिसअपियर नहीं होने वाली.
Yes.
शलभ जी.
तो मैं भी मैम को सेकंड करूंगा इधर ही कि न्यूट्रलाइजिंग एंटीबॉडी टेस्ट इज इम्पोर्टेंट इफ यू वांट टू असर्टेन द इम्यूनिटी. एंड दूसरी चीज, अगर आप प्लाज्मा डोनेशन के लिए भी प्लान कर रहे हैं तो ये वाला टेस्ट जरूर कराइए, बिकॉज ये आपके प्लाज्मा में कितना दम है, न्यूट्रलाइजिंग एबिलिटी कितनी है, वो डिटरमाइन करेगी. और तीसरी चीज इवन सरकार के लिए भी कि इफ यू वांट टू असर्टेन द हर्ड इम्यूनिटी, द ट्रू हर्ड इम्यूनिटी, देन न्यूट्रलाइजेशन टेस्ट इज द फाइनल आंसर. So thank you from my side. Thank you, sir. Thank you, ma'am.
Thank you, Dr. Agarwal. Thank you very much.
स्पेसिफिकली अगर आप किसी वीआईपी को एक इनॉग्रेशन के लिए बुला रहे हैं और आप लिखेंगे कि स्टेज पे 4 लोग बैठेंगे, चारों के न्यूट्रलाइजिंग एंटीबॉडीज पॉजिटिव है, तो वीआईपी को भी अच्छा लगेगा, सेफ लगेगा, वो टाइम दे देगा, नहीं तो बोलेगा कि मैं वर्चुअली इनॉग्रेट करूंगा. So
absolutely, yeah. And the way forward, I feel this will instill confidence in us that when we will be finally get rid of this mask and all.
अगर आपको लगा कि ये प्रोग्राम वर्थ लिसनिंग है, तो इस वक्त में द बेस्ट यू कैन गिव ए गिफ्ट टू योर कलीग इज टू सेंड ए लिंक. किसी का डर मिटा देगा, किसी की जान बचा देगा, किसी का फोबिया हटा देगा. That's all for today. Let me thank Dr. Vandana Lal and Dr. Salab Malik to be here. We'll be back tomorrow 7 p.m. with another show. Keep watching Medtalks with Dr. KK. Thank you and namaskar.
Thank you.
Thank you.